Today I'm going to talk about the work that we have done in the past few years on trying to target a couple of hallmarks of aging in order to reverse age-related cardiac dysfunction in mouse model. And I'll begin the talk by giving a brief introduction on cardiac aging, and then I will go into the first hallmark that we target, which is mitochondrial dysfunction, and we try to target that by a peptide drugs called SS31. And then in the second half, I'm going to talk about another hallmarks of aging, deregulate mTOR signaling, and we try to target that with drugs called rapamycin. And as most of you already know, the prevalence of cardiovascular disease increased with age. And if you look at this graph from American Heart Association, you can see how sharply the prevalence of cardiovascular disease increased with age in our population. And in individuals that are 60 to 79 years old, their prevalence of cardiovascular disease is about 70%, and it gets up to over 80% for individuals that are over 80. And in addition to increased prevalence of cardiovascular disease, aging also leads to deterioration in structure and function of the heart in individuals that don't have any overt cardiovascular disease. In fact, if we looked at different phases of the cardiac cycle, if we look at systole or systolic function or ventricular contraction, it's actually relatively preserved with age. So what that means is if we measure fractional shortening or ejection fraction, which represent the contractility of the heart in systole, these parameters, they are only slightly reduced with aging. But on the other hand, if we look at diastolic function, which is a measurement of the ability of the heart to relax and refuel, this function is impaired by aging. And ventricular filling is comprised of two phases: the early phase, which is attributed by ventricular relaxation, and then the late phase, which is attributed by atrial contraction. So what happened with aging is that due to the impaired relaxation of the myocardium, the first phase, the early phase, is impaired, and the contribution of this phase reduced with age. And relatively, the contribution of the second phase, the atrial, that is due to atrial contraction, increased. So if we measure by pulse rate Doppler or by tissue Doppler to measure the early to late filling ratio, which is E prime to A prime or EA-AA ratio, this ratio actually declines with age. And in between contraction and filling, there's also two isovolumic phases that allow the change in pressure to happen. And these two phases both are prolonged with aging. So if we measure myocardial performance index due to the prolonged isovolumic phases, this index, which measures the time that is spent in these two phases relative to ejection times, also increase with age. So increase in this index is meaning that there's reduced myocardial performance. So increase in index is actually a bad sign. And structurally, we also see ventricular, left ventricular and left atrial hypertrophy with aging. The changes in cardiac function in healthy adults have been documented in two population studies, the Framingham study and the Baltimore longitudinal study on aging. Both of these studies look at changes in cardiac function and structure in healthy adults, and they've shown that there's an increase in LV wall thickness, which indicate hypertrophy, cardiac hypertrophy with aging. They also discover cardiac fibrosis increase with age. And functionally, when they measure E to A ratio, both of these studies show that E to A ratio decline with age in healthy adult. And what they've shown is when an individual is 60 or older, the E to A ratio, the average actually decreased to below 1. When this ratio decreased to below 1, that means the late phase actually contribute more to ventricular filling than the early phase due to relaxation of the ventricles. And at this point, this individual is diagnosed to have diastolic dysfunction. And for individuals that have diastolic dysfunction, they can develop heart failure phenotypes, even if they have preserved or normal ejection fraction. And so if anyone develop heart failure phenotype, they have ejection fraction that are over 50, or what we call normal ejection fraction. They are diagnosed to have HFpEF, heart failure with preserved ejection fraction. HFpEF became an increasingly important clinical problem. If you look at the graph here, you would see that for the percentage of heart failure patients that get hospitalized, and it's actually, if you look at the percentage, the one that have normal ejection fraction over 50, actually is on the rise, is increasing. The proportion that have reduced ejection fraction, the red line here, or the red circle here, is actually decreasing. And those are the heart failure patient with reduced ejection fraction. So that's on the decline versus the increasing trend for HFpEF. And the increase in HFpEF is likely due to the increasing age of our population, because HFpEF is highly prevalent in elderly. And another reason is that currently we don't have any effective treatment for HFpEF, which makes it very important to have a better understanding of the mechanism that lead to diastolic dysfunction or cardiac aging. And so we can develop intervention to treat this phenotype or treat this disease. In order to understand the mechanism of cardiac aging, many aging studies will use animal models, and rodent model is very widely used, and in particular mouse model is widely used in cardiac aging study. The phenotypes of cardiac aging in mouse closely recapitulate what happens in human. And I'm going to talk about the phenotypes in the next slide. And also, because mouse has a relatively short lifespan, it makes aging study in mouse relatively manageable in terms of time. And also the availability of different genetic model, transgenic or knockout mouse model, and different tools that are available make a mouse a good model for this study. And while the cardiac aging phenotype of mouse and human are very similar, one thing is different is that mouse don't develop hypertension or elevate blood pressure with aging, and they also don't have an adverse glucose or lipid profile with age. So when we looked at cardiac aging in the mouse, most of the changes that we see are intrinsic to the change in the heart, but not due to change in the vasculature or the blood pressure or these lipid or glucose profile. And one thing we should consider when we read literatures in these cardiac aging study is that different study oftentimes use different age as their old mice. So we should consider the age group and also the strain of the mice they use when we compare different studies, because that can affect the outcome. So what are the characteristics of cardiac aging in mice? As in human, mouse also have decline in diastolic function with aging. So the graph here, we are looking at different age group of mice from young, 4 to 6 months old, all the way to mice that are older than 24 months old. So what most study would consider old mice. And when we look at diastolic function, E prime, A prime, or EA-AA ratio here, you can see this ratio declines with age. There's actually a 47% decline of E prime, A prime ratio or diastolic function with age. And the next one is myocardial performance index. So this index increased with age in human, and it also increased with age in mice. And this is due to the prolonged isovolemic phases. And mouse also develop cardiac hypertrophy over aging, and can be indicated here by normalized heart rate increase when the mouse get older. And they also have cardiac left atrial hypertrophy. The atrial size actually also increased. And if we look at systolic function, this is ejection fraction here. There's a slight decline, about 12% decline over from young to old in mouse model. So there's a slight decline that is relatively small compared to decline in diastolic function. And another phenotypes of cardiac aging is cardiac fibrosis, and that also happened in mouse model. And this is a Picker series red staining, and the red color here is staining for collagen, and you can see that increased deposition of collagen with age in the myocardium. And to assess cardiac aging in mice, we need some assessment tools so we can use to evaluate whether we see impaired cardiac function and whether we see improvement when we treat this animal. And one of the method that we use is echocardiography. There's different type or different mode of echocardiography. One is the two-dimensional echocardiography, which can be used to measure systolic functions. We can measure fractional shortening, which represent the contractility of the heart. And we can also use a pulse rate Doppler to measure blood flow velocity of the heart, or tissue Doppler imaging to measure tissue velocity, the myocardial tissue velocity. And both of these can measure diastolic function parameter, can measure myocardial, and pulse rate Doppler can measure myocardial performance index. The advantage of using echocardiography is that this is a non-invasive method of measurement. That means we can perform longitudinal assessment on the same animal over time. We can follow the same animal from young to old and look at the changes within the same individual. And this is just a couple images that from pulse rate or from tissue Doppler showing what a normal diastolic function looks like compared to one with diastolic dysfunction. And another assessment we can use to measure cardiac function is to measure pressure volume hemodynamics by cardiac catheters. And this is also a very widely used measurement to measure diastolic function. The disadvantage or the limitation of using this measurement in mouse model is that due to the small size of the mouse and the technical difficulty, this procedure can only be done as a terminal procedure, meaning that we cannot use it to follow the same animal over time. It can only be used for cross-sectional study to compare different animals at the time of at the endpoint. So with these assessment tools, we can evaluate whether we can use intervention that target different hallmarks of aging to restore cardiac function in old mice. And the first intervention or the first hallmarks that we will target is mitochondrial dysfunction. So the figure here is a summary of different cellular and molecular changes that numerous aging studies have shown to be changes during aging. So this review article basically summarized all the changes that have been published with that change with age and may contribute to the aging of the organism as a whole. And these hallmarks include telomere attrition, epigenetic alteration, loss of proteostasis, and the one that we want to focus on in the first part of the talk is mitochondrial dysfunction. In 1956, Denham Hammond proposed the free radical theory of aging. So in this theory, he proposed that reactive oxygen species, ROS, accumulate with age and create oxidative damage on macromolecule inside the cells. And this macromolecular damage will accumulate and lead to organ damage, dysfunction, and this will drive aging. And based on this theory, when we scavenge ROS, when we scavenge the reactive oxygen species, we should delay aging. However, a few studies have shown that when you overexpress antioxidant protein, which will scavenge ROS, scavenger reactive oxygen species, they fail to see an increase in lifespan. And this leads to a modification or an update of the theory to become the mitochondrial theory, free radical theory of aging. So this update theory proposed that the location, the subcellular localization of the ROS also matters, and mitochondrial ROS is the driving force of aging. And this is supported by this study published in 2005 by the Respiration lab. And what they did is they overexpressed a human catalase, which is an enzyme that can convert hydrogen peroxide into oxygen and water. So it will scavenge the ROS. And when it's targeted to the mitochondria, this enzyme will specifically scavenge mitochondrial ROS. And the result of that is that they see an 18% increase in lifespan in this animal that express mitochondrial-targeted catalase. And importantly, when they overexpress this enzyme in peroxisome or in nuclear, they don't see a similar lifespan extension, which suggests the location of the ROS scavenging is important, and mitochondrial ROS is the driving force of this lifespan limitation. They also followed in a follow-up study, looked at cardiac function in these mice. And what they see is that compared to wild-type animal, which is shown in the black line here, mCAT animal, which have scavenger, which have reduced mitochondrial ROS, have attenuate decline in diastolic function. Also, they have reduced myocardial performance index, which suggests that they have improved myocardial performance compared to wild-type animal. They also have reduced cardiac hypertrophy, so their normalized heart rate is lower when compared to wild-type animal. So this supports that scavenging mitochondrial ROS is protective in cardiac aging. One limitation with transgenic animal is that the ROS scavenging happens throughout lifespan. And this is not what usually a drug's target, what we hope for a drug target. We want to see whether we can reverse the phenotypes or the dysfunction when it already happened, because that's what most of the time, that's what we can usually target for treatment. And in order to do that, what we try to do is to use a pharmacologic compound, SS31 peptide, which has been shown to be protective in some mitochondrial dysfunction model, to see whether we can use this compound and mimic what we see in mCAT mice to improve mitochondrial function and reverse pre-existing cardiac aging in mouse model. So the reason we choose SS31 peptide is because we have studies have shown that it have similar protective effects as mCAT mice in some cardiovascular disease model. What we're looking at here is the structure of SS31 peptide. So it is a tetrapeptide, so only 4 amino acids, and it can be targeted to the mitochondria. So when this drug is given to the animal or an individual, it's highly concentrated in the inner membrane of the mitochondria. And the reason for that is likely because this molecule will bind to cardiolipin, which is the phospholipid that is exclusively in the inner membrane of the mitochondria. And by binding to cardiolipin, this compound has been shown to promote mitochondrial membrane curvature and enhance ATP production and reduce cytochrome C peroxidase activity. So in this study, 2011 study in JACC, what they see is if you give animal 4 weeks angiotensin, so normally after 4-week angiotensin treatment, there will be diastolic dysfunction. So the E/A ratio after 4 weeks is reduced in control animal. But in animals that were given SS31 peptide, or in animals that have an inducible mCAT, both of these treatment will increase diastolic function. And both of these treatments also are similarly protective in another model of cardiac dysfunction, which is TAC-induced heart failure, so transverse aortic constriction to induce heart failure. So in this model, it has been shown that both mCAT expression and SS31 is able to reduce heart weight, so cardiac hypertrophy, or lung weight, wet lung weight, suggesting of pulmonary edema. Both of these is attenuated by the treatment. And both of these treatments also similarly improve systolic function. And because of the similar protective effects of SS31 and mCAT that has been shown in this cardiovascular disease model, we want to ask whether we can use SS31 treatment to reverse pre-existing cardiac aging in mouse model. So what we did is we have young and old C57 black 6 mouse, and we used osmotic mini pump to deliver SS31 peptide to this animal and compare them to saline control. We followed their cardiac function at 4 weeks and 8 weeks, and also looked at exercise performance in these mice by treadmill running. So when we looked at the the function of the heart, what we observed is that for diastolic function, so this is E to A ratio, E prime to A prime ratio of these mice at baseline, which is 0 weeks here. The ratio is close to 1, which is decreased compared to a normal young animal, which usually we observe by our method is about 1.5. So this decline in diastolic function is normalized or is improved over the course of the 8 weeks treatment in the group that is given SS31 peptide. And when we looked at myocardial performance index, so this index increased with age because of the prolonged isovolemic phases. And usually in our mouse, on our hand, this ratio is about 0.3 to 0.4, and is increased in these aging mice. And this ratio is progressively reduced in only in the group that is given SS31. So other than the improvement in cardiac function that I've just mentioned, these mice also have attenuated cardiac hypertrophy. So in the old mice that is given SS31, the normalized heart rate is reduced compared to old control animal. And because cardiac function is closely related to exercise tolerance of an animal, we look at their treadmill running performance. So with age, the animal run a shorter time on treadmill, and that is improved after we give 8 weeks of SS31 to this animal. And then we want to understand the mechanism of how this SS31 action is mediated. The first thing we looked at is mitochondrial function, because that's what we're trying to target with the peptide. And to do that, we isolate cardiomyocytes from animals that are young, old, or old mice that are treated with 8 weeks of SS31. So in these isolated cardiomyocytes, we measure their cardiac function by performing CHOS analysis using a mitostress test protocol. So, and this is just an average trace that we see with the 3 different groups. So the key changes that we noticed with aging is that these cardiomyocytes have increased basal respiration. And this increase in basal respiration is attributed by an increase in proton leak, which is the phase that we measure here. So, and both of these increase in basal respiration and proton leak is normalized or is reversed when we give animal SS31 for 8 weeks. So SS31 is able to reduce age-related increase in proton leak in cardiomyocytes. The next thing we look at is whether we see reduced oxidative stress or reduced ROS production in these cardiomyocytes. And we isolate isolate cardiomyocyte, we use fluorescent indicator, mitoSOCs, to look at superoxide production. And as you can see here, the superoxide production in old mice treated with SS31 is reduced compared to control animal treated with saline. And we normalize the signal to mitotracker green, which just normalized for mitochondrial content. And this is just a graph showing that mitochondrial superoxide production is reduced in SS31 treated animal. Next, we looked at mitochondrial hydrogen peroxide using a different dye, MitoPY1, and similarly, we see reduced MitoPY1, and after normalization, the signals of MitoPY1, a normalized MitoPY1 or mitochondrial hydrogen peroxide production is reduced in the group that is given SS31 for 8 weeks. And because of this reduced ROS production, we also looked at whether ROS oxidative modification or oxidative damage in protein is reduced by SS31. So one way we looked at that is by a proteomics approach. We use proteomics to look at protein S glutathione level in heart that are young, old, or old heart that have been given SS31 and for 8 weeks. And what you're seeing here is the histogram of the result of log2 fold change of glutathione. So anything that is on the right-hand side here that is above 0, meaning that there's an increased level in either old compared to young, or for the red in old SS31 compared to the young. So the most of the blue bar are on the right side of the histogram here, on the right side of 0, suggesting that there's overall an increased level of glutathione in old heart, old control heart compared to young heart. And when we give animal SS31 for 8 weeks, we shift this glutathione level closer to 0. So it's the shift to the left, and suggesting that the peptide treatment is able to reduce glutathione. And another oxidative modification we look at is protein carbonylation. And compared to young animal, old animal has increased level of protein carbonyl in myocardial protein. And this increase is normalized when we give animal 8 weeks of SS31. And because we observe improved diastolic function in this animal, we try to understand what are the changes that could mediate these changes in diastolic function. And one key regulator for diastolic function is myofibrin protein phosphorylation. So we investigate whether we see any changes. So with age, there's a decline in phosphorylation of myosin binding protein C, MyBP-C, and this decline is normalized when we give 8 weeks SS31 to mouse model. And this decline or reduced MyBP-C phosphorylation have been linked to impaired diastolic function. So this normalization could in part contribute to the improved diastolic function that we observe. And then we also look at troponin I phosphorylation, but we don't see any difference that is mediated by SS31 treatment. And then other regulator of diastolic function is the stiffness of the myocardium, and titin, sarcomeric protein, can regulate the stiffness of the myocardium by because of its altered compliance. There's 2 isoforms of titin, and the isoform shift of titin can alter the stiffness or the compliance of titin. But we don't actually, we did not detect any changes in titin isoform after SS31 treatment, suggesting that the mechanism of the SS31 action is independent of titin isoform shift. And because we want to evaluate the persistence of the SS31 intervention. So we did this experiment where we give mice 8 weeks of SS31, and then we remove the mini pump and stop the treatment and follow cardiac function afterward. So what we see is after 2 weeks of stopping the treatment, the diastolic function, E to A ratio of this the SS31 treated group still remain significantly improved compared to control, but at 12 weeks, we are starting to lose the benefit. So this suggests that the benefit of SS31 in diastolic function is persistent for about 2 to 4 weeks after we stop the treatment. So in summary, in this first part, we observed that SS31 treatment of 8 weeks can reverse cardiac aging phenotypes in mouse model. And I don't have time to go into detail, but in this study, we also compared the effect of SS31 with mCAT overexpression given by adeno-associated virus. So we only overexpress mCAT in old mice after they develop pre-existing cardiac aging phenotypes. So both of these treatment, we observed reverse cardiac aging phenotype, improvement in diastolic function. And the mechanism that SS31 is working, we show that this is related to reduced mitochondrial ROS production and also reducing mitochondrial proton leak in the aged cardiomyocyte. And this is consistent with a reduced level of protein oxidative modification that we observe in myocardial protein in the SS31 treat part. And the benefit in diastolic function could be mediated by changes in myofibrin protein phosphorylation, in particular, the reduced age-related decrease in MyBP-C phosphorylation that is restored when we give animal SS31 peptide. So before I go to the next, okay, so now I'm going to talk about the second hallmarks of aging that we try to target. And the hallmarks is deregulate nutrient sensing. And in particular, we are focused on mTOR signaling, which is a nutrient sensing pathway. And we try to target that with a compound called rapamycin. And in the aging research field, the most studied, most well-established intervention that have been shown to increase lifespan is calorie restriction. In multiple animal models, from mouse, fly, worms, and yeast, it has been shown that calorie restriction can increase lifespan. And it is believed that one of the key mechanisms that calorie restriction mediates its benefit is by inhibiting mTOR signaling. So this is just a diagram of the mTOR signaling pathway. So when nutrients is abundant, nutrient signal will activate mTOR one of the mTOR signaling, the TOR1 of mTOR signaling branch, and then it will lead to increase or enhance in protein translation through 4EBP and S6K, and it also will inhibit autophagy when mTOR is activated. So during calorie restriction, when nutrients is limited, mTOR is inhibited, and then protein translation will be inhibited and autophagy will be promoted. And rapamycin is a compound first found in the soil bacteria in the Eastern Island. And this is a compound that has been shown to inhibit TOR1 complex and is considered as a mimetic of calorie restriction because of that. And the intervention testing program of NIA has tested the effect of rapamycin on lifespan of in mouse model. And what they have shown is that in both male and female mice, rapamycin is able to extend lifespan. So because of that, we want to see whether rapamycin and calorie restriction can also reverse pre-existing cardiac aging phenotypes. And back in 2014, we published the data of the study. We show that when we give old animals that are 25-month-old at baseline at the beginning of experiment, 10 weeks of either calorie restriction or rapamycin, we are able to reverse different phenotypes of cardiac aging. We see improvement in diastolic function, improvement in myocardial performance, which is a reduction in their myocardial performance index. And we also see reduced cardiac hypertrophy. And the phenotypes are similar between treatment with rapamycin versus with calorie restriction. And when we try to understand the mechanism, one of the study that we perform is a proteomic study. And this is a heat map of the result that we find, or simplified version of the heat map. And basically what you're looking at is for the 3 different comparisons. So young control versus old control on top, and then old calorie restricted or compared to old control, and then old rapamycin treated versus old control. And whenever you see red here, it means there's an increased ratio, which means there's higher level of this protein in young, in calorie restriction, or in old rapamycin treatment compared to old control. And when you see blue color, that means it's reduced or it's lower in young, in rapamycin treated, or in calorie restriction compared to old. So what we see is that with age, there's an increased, there's a decreased level. So young is higher than old for mitochondrial dysfunction or mitochondrial, basically mitochondrial protein related to mitochondrial function, fatty acid oxidation, and TCA cycle protein. So this mitochondrial protein level is higher in young, it's higher in animal treated with calorie restriction or rapamycin. And on the other hand, when we look at protein that are related to glycolysis or glucose metabolism, they are blue here, meaning that they are lower in young, in calorie restriction, and in rapamycin treatment. So there's a change or a shift from protein that is in mitochondrial function to that we see an age-related decrease in this protein. And then we see an age-related increase in protein in glycolysis. And all this is reversed when we give animal either calorie restriction or rapamycin. So if you look at the comparison in these 2 groups, it's closely resemble what the young control looks like. And because of these changes that we observe with this energy metabolism pathway, we perform a metabolomic analysis to look at whether the metabolite behave in the same level. So this is a comparison of the rapamycin effects. So we compare how 10 weeks rapamycin treatment alter metabolism or this metabolite profile in old hearts. So compared to old control, rapamycin treated heart have reduced level of metabolite in glycolysis and increased level of metabolite in TCA cycle, which agree with what we see with the protein or the enzyme that is part of these pathways. And so if we put it back to the diagram here, what we observe is that with rapamycin treatment, we are basically reversing the increase in glycolysis that we see with age, and we also are able to increase fatty acid oxidation or increase TCA cycle when we give these old mice rapamycin for 10 weeks. So the first study that we performed that was published in 2014 is a 10-week study. So we only look at the changes at the end of the study. That is a snapshot of what is happening at 10 weeks. When we look at some of the changes, that we see this increase in mitochondrial protein, and we see these other changes. But some of these changes that we expect, we actually are not able to see at the endpoint, and which is why we want to go back and look at the changes that happens early on in after we initiate the treatment. And so when we perform our study that looked at both the changes that happens earlier in the treatment and later, we first want to confirm that in this study, we also see changes in diastolic function and in myocardial performance that we see in the earlier study, which we did. And we also show that the improvement in rapamycin-induced cardiac aging reversal actually occurred or begin pretty early on in 2 to 4 weeks after we initiate the treatment. We already see an improvement in diastolic function, and we already begin to see a decline in myocardial performance index. So then performed a cross-sectional study to look at what are the early changes after we begin rapamycin treatment compared to the late changes that we previously observed in 10 weeks, the 10-week study. So we performed a 1-week rapamycin treatment and 2 weeks rapamycin treatment to look at the acute changes. So first, we confirmed that this treatment can inhibit mTOR signaling. And indeed, we see a reduction in S6 phosphorylation, which indicate mTOR1 inhibition, both in early and late time point. And we also look at AKT phosphorylation and PKC alpha phosphorylation, which is downstream of TOR2. And we see inhibition of TOR2 both in early and late time point. And one thing I mentioned that in earlier study, we are not seeing some of the changes we expect with rapamycin treatment. So by inhibiting mTOR signaling, we are expecting an increase in autophagy. But when we looked at heart at 10 weeks after treatment, we actually see no difference in autophagic markers, LC3 or ATG5 in this heart sample. But when we look at the earlier time point, we noticed that there's actually an increase in autophagy induced by rapamycin treatment at 1 week after treatment. But this increase is gradually disappear. So it became back to the baseline level after 2 weeks of treatment. We also perform an autophagic flux measurement and see similar result. And another change that we observe is changes in mitochondrial biogenesis, which also at 10 weeks, there's no increase in mitochondrial biogenesis, even though we observe this increase in mitochondrial protein content by proteomics. And if we look at the earlier time point, there's actually increase in markers of mitochondrial biogenesis. So there's an increase early on that is disappear or went back to normal after 2 weeks of the treatment. So in fact, after this study, after we measured the mitochondrial biogenesis changes, we looked at proteomic changes and find that at 2 weeks after we initiate rapamycin treatment, we already see the proteomic changes that we observed in the 10-week study. So the decrease in glycolysis-related genes or related protein and the increase in fatty acid-related protein actually take place in the first 2 weeks after we initiate rapamycin treatment. And to see if these changes is actually corresponding to a shift in substrate utilization, we perform an isolated heart perfusion using the Langendorf system and use labeled substrate, label either glucose or fatty acid to measure the utilization of this substrate by the heart. And what we see is that with aging, there's a decrease in utilization of fatty acid, shown by the decrease in the black bar. And this is restored after we treat animal with rapamycin, especially in the earlier time point. So overall, what we see with the rapamycin treatment is that it can reverse cardiac aging phenotype in old mice. And when we look at the time course of the changes, autophagy is induced in the first week of rapamycin treatment and then go back to baseline afterward. And if we look at mitochondrial biogenesis, it's increased in the first 2 weeks and then slowly also return to baseline afterward. So most of these remodeling happens in the first 2 weeks of the treatment. But what are the mechanisms that actually mediate the continuous improvement in cardiac function is something that we're trying to understand right now. And we are looking at changes in calcium handling and passive stiffness and try to understand how this earlier mitochondrial remodeling is mediating the functional benefit that we observe later on. And also the early changes that we observe is also consistent with the change in substrate shift that we detect by the isolated perfused heart study. So overall, the take-home message is that we can target hallmarks of aging, and these are potential treatment for age-related diastolic function and HFpEF. And in particular, we look at 2 interventions that target mitochondrial dysfunction and target mTOR signaling, and show that they can reverse pre-existing phenotypes of cardiac aging. And I'll end by acknowledging all these people that helped in the these two projects. So this is my lab, and they're not the one that work on the study that I present, but they are working on the follow-up of these findings. And then the study is performed while I'm a postdoc in Rabinovich lab. And so I want to thank all the people in there. And then my collaborator, many of them are at UW and also at PNNL, and my faculty mentor at OMRF, Aging and Metabolism Department. And thank you all for your attention.
Thank you so much, Dr. Chow. That was a really fascinating talk. I know I learned a lot, and what we will do now is just jump right into our Q&A session. We have about 5-6 minutes. So let's start with our first question. And again, I would encourage everyone to keep on asking questions. We'll answer as many as we can, and then also following the session. Has SS31 been tested in clinical trials for heart failures or other diseases?
Yes, there has been a few clinical trials on SS31, and a couple of them are on heart failure. There's one for heart failure with reduced ejection fraction called Progress HF, and then there's one for HFpEF called Restore HF. The data or the result of the one with reduced ejection fraction has been published. So it's a 4-week treatment, but so the drug is highly well tolerated, so there's no safety concern, but they don't see an improvement in their primary outcome after 4 weeks. The result of the second study, the HFpEF, has not been published. At least the phase 2 and 3 study has not been published. But interestingly, there's another study on BAF syndrome, which is a disease with abnormal cardiolipin, so the target of SS31. That study, they look at different outcome, but one of the outcome that they look at, one of the secondary outcome that they look at is stroke volume. So in that study, it's actually a longer term study. There's 2 parts of the study, and overall the total duration of the treatment is 48 weeks. And I think from what I remember, they see an improvement in stroke volume after 36 weeks of treatment, about 36 weeks of treatment. So I think the lack of improvement that they see with the heart failure with reduced ejection fraction study, which is only 4 weeks, might be due to not enough duration of the treatment, but that need to be further studied. And there's also other clinical trial on one is mitochondrial myopathy, and then there's also clinical trial on SS31 on some eye disease.
Okay, perfect. Thank you. And in our responses in the written transcript, perhaps we'll provide some links to those references as well.
Mm-hmm.
Will combining the 2 treatments, so you talked about SS31 and rapamycin, offer additive benefits?
Yeah, so I don't have any data on that, but my thought is whether combining 2 treatment will have additive effect depending on whether they the mechanism of their action overlaps or not. So when we do the SS31 study, we also have another small experiment that we do overexpression of mitochondrial-targeted catalase by AAV. So when we combine mCAT, AAV, and SS31, which both act on mitochondrial dysfunction, there's no additive effects. But if you combine 2 treatments that target 2 different pathways and mediate benefit through different mechanisms, then I think there's a potential to see additive benefit. For SS31 and rapamycin, I think there will not be a completely additive benefit because some of the downstream mechanism might converge. So for rapamycin, we see some improvement in mitochondrial function. We see this increase in protein expression in fatty acid oxidation, in mitochondrial function protein in this pathway. So I think there's some overlapping with the 2, but if there's potentially there's some area if the mechanism is different, there is potential for additive effects.
Okay, and I guess along the same lines, Dr. Pandey is asking from part 1 if SS31 and mCAT are put together. Would that
Right.
enhance the cardiac beneficial effects, synergistic or additive?
Right. So that's what we did, and we do not detect additive or synergistic benefit. And we think that is because they both act on reducing mitochondrial loss and improving mitochondrial function. So which is what I think when both intervention is targeting the same hallmarks or the same mechanism, then there might not be any additive effect. But if we look at another intervention that mediate the benefit through a completely different pathway, then there are chances that we will see the effects, but not with the case for mCAT and SS31.
I see. Okay, perfect. Since the changes in autophagy and mitochondrial biogenesis occur in the first 2 weeks of rapamycin treatment, will 2-week rapamycin treatment be enough to mediate the cardiac benefits?
Right, that's a very good point. So that's something that we have not looked at, but we want to. So because of the early remodeling in autophagy and mitochondrial biogenesis happens so early on and like return to baseline after 2 weeks, we think if we stop the treatment and follow on the function, there will be continuous improvement of the function based on the benefit of this earlier remodeling. But we might not see the full benefit that we see at the time if we continue the treatment for 10 weeks. If there are additional changes that takes longer to complete or to mediate the benefit. So what we see right now, we only look at 1 and 2 weeks and see these changes in autophagy and mitochondrial metabolism biogenesis. But if we look at some later time point and see changes that takes, for example, 4 weeks or 6 weeks to take place. For example, some changes in extracellular matrix, for example, or some changes in myofilament protein that takes longer to develop. Then continuing the treatment for 10 weeks may, compared to pausing the treatment at 2 weeks, we might see a slightly more benefit. But I do believe only giving the treatment for 2 weeks and stop, we will still see and continue to follow the function, we'll still see some improvement by this short-term treatment. Okay,
perfect. We're at that 60-minute mark, but let's maybe just spend a few more minutes for those that of you that can stick around. Please do. And do you want to maybe look at some of the remaining questions that have been submitted? Are there any that you feel would be important to answer while everyone is still online?
Yeah, let me go through the Q&A box. Let's see. So this few questions from Debbie. Let's see. Will rapamycin treatment reduce free radical in myocardium of aging mice is one of the questions he asked. And we actually we didn't look at ROS production, but we looked at oxidative modification of the myocardium in rapamycin treated mice. So we have looked at protein carbonylation, and we see reduced protein carbonylation in the myocardium of rapamycin treated mice, which would suggest that there could be reduced free radical in the myocardium after rapamycin treatment.
Okay.
And let's see. We have a question on ventricular hypertrophy. Can ventricular hypertrophy be reduced by free will running compared to treadmill, since the latter may be stressful physiologically? So we only perform treadmill running to test whether exercise performance of the animal is improved. We didn't perform treadmill running during the course of the treatment. So that's actually something we measure at the end point. Whether free will running will improve or reduce cardiac hypertrophy, I'm not sure, because exercise can also induce physiological hypertrophy. So it's not pathological, but exercise can also induce hypertrophy in, but not in an impaired function environment. So I'm not sure what free will running will do in this case, but we are not trying to, we're not doing treadmill running for multiple times. We only do it at the endpoint. It's like a 1 week protocol that we train the animal to accommodate the treadmill and then measure. So it shouldn't have any stress during the 10 weeks of the treatment or the 8 weeks of the treatment that we give SS31.
Perfect. Okay, and maybe why don't we answer one more? There was also a question that came in beforehand, and that is, what will happen after you stop the rapamycin treatment? Will the function stay improved, or will it decline? Perhaps that's a question to tackle.
Yeah, so that's a good question. It's actually a study that the Rabinovich lab published in 2020, or 2020. So this year, there's we published a paper that so after giving rapamycin treatment in these mice, we stop the treatment and follow cardiac function in these mice. So we follow after, so in that study is 8 weeks rapamycin treatment, and then after stopping the treatment is another 8 weeks of follow-up. So at 16 weeks, which is 8 weeks after stopping the treatment, there's still a significant improved cardiac function in these mice compared to the control group. So the persistence of the rapamycin treatment is actually, in that sense, for diastolic function at least, is better than SS31. And so what we think is when you give this treatment for a period of time, like 8 weeks, 12 weeks, 10 weeks, you kind of reset the equilibrium. So you bring the heart back to a better baseline. And then when you stop, it will start to decline, but it won't quickly return to normal. So it takes time. And depending on the mechanism of the benefits, some might be more long-lasting. Like changes in matrix could be more long-lasting if it's something related to energy production, ATP, things like that, that might be more transient.
Okay, perfect. Well, thank you so much for those thorough answers. We're going to let everybody get on with their day. Thank you so much for attending. If anyone has questions for Dr. Chow, her email is listed on the slide. It's ann-chow@omrf.org. My email address for anyone that wants to reach out to me, if there's any technology questions, it's mhess@syntaga.com. So Dr. Chow, thank you so much for being with us today, for taking the time to prepare this presentation. We really appreciate it. And thank you to everyone in attendance for taking time out of their day for being with us today as well. So
thank you.
Yeah, you're very welcome. Thank you again, everyone, and enjoy the rest of your day.